Okay, let's talk about aging. What if I told you it isn't just about things breaking down? What if it's not random wear and tear, but an actual program running in the background of your cells, a kind of genetic ghost in the machine? Well, a really groundbreaking new study suggests aging is exactly that, a biological program that gets hijacked.
So yeah, that's the big question, right? For the longest time, the main idea has been that aging is just an accumulation of random damage. But what if it's more deliberate? What if the very same instructions that guide our development from an infant to an adult just kind of keep running? In this explainer, we're going to unpack the evidence for that very idea.
You know, we tend to think of our lives in two distinct phases. We grow up, and then we grow old. But what if that whole idea is wrong? Researchers are now finding that maturation and aging aren't two separate things at all. Nope, they're like two acts of the very same play, driven by the exact same molecular machinery.
To really get a handle on this genetic hijacking, we first need to check out the crime scene, the inside of our cells. We have to look at how our genetic instruction manual is organized, and, more importantly, how it's read over a lifetime.
So get this. Every single cell in your body has the same massive instruction manual, your DNA. But it's not just a bunch of loose pages. It's wound up super tightly into a structure called chromatin. Now the key here is that not all pages of this manual are open at once. Which instructions are available and which ones are locked away, well, that determines everything about what a cell is and what it does.
All right, so if chromatin is the instruction book, what decides which pages to read? Well, that's the job of proteins called transcription factors. You can think of them as molecular bookmarks. They literally latch onto specific spots in the DNA and tell the cell, hey, read this page. We need this instruction right now. They are the ultimate shot callers for a cell's identity.
In our story of genetic hijacking, there are three main groups of these transcription factors that are absolutely crucial. So, let's meet our three main suspects.
First up, we've got a family of transcription factors called AP1. We're calling this one the instigator. What the research shows is that as we get older and as our cells experience stress, AP1 activity just goes up and up. Its main job? To find and open new pages in that genetic manual, pages that were previously locked shut.
Our second suspect is a group we'll call the guardians of youth. These are cell identity transcription factors. Their whole job is to stay put, latched onto the pages that define a cell's original specialized function. You know, keeping a skin cell acting like a skin cell or a neuron acting like a neuron. They're all about maintaining the status quo.
And finally, we have the stabilizer, a factor called CTCF. Now, while the other factors are in this dynamic push and pull, CTCF is the anchor. It locks down the most essential, non-negotiable instructions. Think of it as the core operating system of the cell that has to stay active and stable no matter what.
So, we have our suspects, the instigator, the guardians, and the stabilizer. Now let's see how they all interact over a lifetime in a process the researchers have brilliantly named the siphon model. This, my friends, is the heist.
So here's how it all plays out. When we're young, the guardian factors are in total control. But over time, the instigator, AP1, starts opening up more and more of these new genetic sites. And this is where the competition begins. These new sites act like powerful magnets, literally siphoning resources and other transcription factors away from the original youthful sites. And as those resources get pulled away, the old sites shut down, the cell's function changes, and the active process of aging kicks into high gear.
It's a really compelling story for sure, but how did the researchers actually prove this genetic hijacking is happening? Okay, let's dive into the evidence.
First off, the sheer scale of this research is incredible. The scientists didn't just look at one or two cell types. No, they analyzed more than 45 different cell types from both mice and humans, spanning the entire life cycle from development to old age. This gives their conclusions an enormous amount of weight.
And across all those different cells, they saw the exact same pattern. You can see the shift right here. In youthful cells, the guardian sites are buzzing with activity. In age cells, they've gone quiet. Meanwhile, the instigator sites, which are pretty quiet in youth, become highly active as we age. It's a predictable, system-wide changing of the guard.
The researchers themselves summed it up perfectly. The very process that drives our healthy development, this AP1 opening up new genetic territory, well, that's the thing. The problem is that process doesn't stop. It just keeps on going throughout our lives. And this ongoing opening of our DNA is essentially hijacked, becoming the very engine that drives aging.
When you put all this evidence together, it leads to a radical, and I mean radical, new way of thinking about why we age. It's a fundamental shift in our understanding.
So let's recap the big takeaways here. First, aging isn't a separate thing. It's just the tail end of the exact same program that drives our growth. Second, this means aging is an active, genetically driven process, not just a passive pileup of damage. Third, the whole mechanism is this predictable siphoning effect, where a shift in transcription factors remodels our cellular instructions over time. And finally, this isn't something that just kicks in when we're old. It's a continuous program that starts early and unfolds across our entire lives.
And that, of course, brings us to the final, absolutely fascinating question. If aging isn't an inevitability of physics, but is actually a feature of our biological software, does that mean that one day we could learn to rewrite the code?